Okay, so let's make a drawing here of a general eukaryotic cell. I'm going to start off by drawing kind of a big oval, a big circle. I'm going to leave a little space to write around the outside. That's going to be the cell membrane, but I'll come back and label it near the end because there's going to be a lot of things to show in this diagram. Next, I'm going to draw a circle within the circle, and that is going to represent the nuclear membrane. And then I'll sort of put it off center so we leave space to draw a dot, a dark dot within the circle. That is going to represent the nucleolus. And then, of course, we need some DNA in the nucleus. I'll show that with a bunch of squiggles. And then there's some things called nuclear pores that are going to allow material to come in and out of the nucleus. And this whole thing here with all those parts we just drew represents the nucleus of the cell. So we got a nucleus with a nucleolus DNA. We're showing some nuclear pores. And next, we want to sort of strategize here a little bit. I think it's important to draw some of the larger structures inside the cell first. So one of these is the endoplasmic reticulum, the ER. And if you look at a map with a whole bunch of highways connecting cities, it's like a system of highways that are connecting all the cities. Now, the endoplasmic reticulum is everywhere, but we can't really draw it everywhere because there's a lot of other parts in the cell to draw. So I've drawn sort of two sections of endoplasmic reticulum showing two different types. Just the one here, I'm going to put all these little red dots on the endoplasmic reticulum, and these red dots represent ribosomes. Each of these little red dots is a ribosome. They're really tiny, too small to see with a microscope. They are studding like these little bumps. They're like little spheres. They're all over the endoplasmic reticulum, some of the endoplasmic reticulum. Now, there can also be ribosomes just floating around, not on the ER. We might refer to those as the free ribosomes, more red dots. They're just not on the endoplasmic reticulum. Now, the endoplasmic reticulum that has the ribosomes, because it's bumpy and rough and studded with ribosomes, we're going to refer to it as the rough ER. And then there's a lot of other ER that doesn't have ribosomes. We refer to that as the smooth ER because it's smooth. It's not studded with these little bumps on it. Another good thing to draw, like sort of a stack of pancakes that's a little bit bigger. In the middle, we have the Golgi apparatus, sometimes called the Golgi body. In the interest of space, I'm going to call it the Golgi body here. It's a pretty big thing that you'll see in a microscope if you look at a cell. And another kind of big thing, we'll talk more about this and why it's so big, is something called, a mitochondria. And there would, of course, be more than one of these mitochondria. There might be another one down here. So we've got the endoplasmic reticulum with and without ribosomes, the Golgi body, the mitochondria. These are some of the bigger structures in the cell. I might just point out here before we get going too much further that the white stuff in the background, excuse me, we almost lost it there for a second while I was trying to change the pen. The white stuff in the background. I'll write it in blue, but all the white stuff in the background is the cytoplasm, like the water in the cells. So those free ribosomes, the mitochondria, are sort of floating around in the cytoplasm, if you will. We have some smaller structures then to draw in the cell. Occasionally, there's just like an empty little cave. We refer to that as a vacuole, and there, be various vacuoles. I'll maybe just show two of them here. And we have a thing, I'll draw it as a dark little circle that represents a lysosome, and I'll maybe show another lysosome but not label it, and another little structure, another little organelle. I'll maybe show this one with some dots in it so we can distinguish from the lysosome is a peroxisome. And again, there would, be various peroxisomes within the cell. So I think we've got most of the major parts here, although there is one more part that I'd like to draw in. We have something called a centriole. I can maybe show a couple. Those are like tubes. And I'll draw these little sticks. These are the pieces that the centriole is made of. Those are the microtubules. That's one word, but I'm going to hyphenate it because it didn't quite fit there. So that's all the basic organelles on the cell. We need to go back and label what we did first. The cell membrane, right? Like I said, the diagram is a little full, so I have some space conveniently left in the lower left there to show the cell membrane. And just like the nucleus has nuclear pores, the cell membrane is going to have individual pores, also known as channels, that are going to let material in and out of the cell. So there you have it. That's a drawing of a eukaryotic cell in about 7.5 minutes.